hit send. Keep the drop-down option as all panelists. Today's presentation is how to demonstrate virtual bioequivalence for complex dermal generic drugs. Today's speaker is Dr. Sebastian Pollock. Dr. Pollock is currently a senior principal scientist at Certara, where he leads the development of the cardiac safety simulator. He's also a professor of biopharmaceutics at the Faculty of Pharmacy at the Jagiellonia University in Krakow, Poland. Sebastian has published over 50 peer-reviewed articles and holds a doctorate, and a master's degree from the Faculty of Pharmacy at the Jagiellonia University Medical College. Bastek, I'll now turn it over to you to begin the presentation.
Thank you very much, Suzanne. Appreciate. Hello, everybody. Good morning, good afternoon. It's a great pleasure to be here, and thank you very much for joining, especially now in this pretty dynamic situation connected with the spread of the disease. So thank you very much. I'm really looking forward to this presentation. I'm really looking forward to the Q&A session. So if you had any questions at the end of the presentation, I'll be happy to take them. By the way, the picture which you see here is pretty old. I wish I could be like that, but I decided that when I turn 40, I won't change my picture anymore. So it's pretty historic. What is the agenda for today? General introduction about the generics. Why it is important and to what degree it can be covered by a novel concept which help us to introduce new drugs to the market. Then a general idea of the virtual bioequivalence, and generally it is part of the model-informed drug development strategy or paradigm. So I would like to spend a short period of time on that as well. Then I will describe briefly the model which we use for the dermal drugs. I will cover development, verification, and utilization very quickly, and I will give you one or two case studies to describe the general concepts, how it can be used. This webinar in general is one of the reasons why we are having this webinar is our recent success in applying virtual bioequivalence study in the dermal area and the agency approval. That's something what we are very proud of, and that's something what proves that the general concept of virtual bioequivalence makes sense, and it's scientifically sound and can be used. So that's general in general the starting point of the whole discussion. As I mentioned previously, the virtual bioequivalence concept is part of the wider paradigm or wider idea, which is called model-informed drug development. MIDD is a quantitative framework that's absolutely critical here. It's a quantitative framework which is based on the integration of data, knowledge, models, and the ultimate aim is to improve the quality, efficiency, and cost effectiveness of the decision making. In general, what we aim for is elimination of unnecessary clinical trials or unnecessary burden, which is connected with the preclinical and clinical trials. All the regulatory bodies around the world, including FDA, EMA, PMDA, and others, identified IMDD as an important pathway which can help to lower the drug attrition and to help dealing with the regulatory uncertainties. And this is exactly where virtual bioequivalence fits, what I will try to prove or describe during today's presentation. Certara is a leading provider of MIDD in general and regulatory science. And MIDD is used across the company for various projects as part of the development cycle for both novel and generic drugs. For each and every element which is important for these compounds or drugs, including tox assessment, safety, efficacy, dosing, or product formulation. And this formulation, by the way, will be one of the most important elements of this presentation and the virtual bioequivalence concepts. Why is that? Because evaluation of the bioequivalence for topical products is a modular approach, which means that it is, or it can be based on different elements, beginning from those which describe the excipients or constitution of the drug, where the drug is API plus formulation, obviously, up to the probably most relevant, but at the same time most challenging in vivo studies. So it can be based on a Q1 and Q2, which is qualitative and quantitative sameness. Q3, which means physical and structural similarity. IVRT and IVPT, which is in vitro release and in vitro permeation test, or combination of all of them. At the same time, there are other ways which allow us to evaluate bioequivalence, and this includes in vivo pharmacokinetic and pharmacodynamic studies and in vivo comparative clinical endpoint studies. The latter is most advanced, but at the same time most challenging, especially for some of the drugs, where the clinical endpoint is subjective, and that can make this study very uneasy to perform and results challenging to analyze. In silico, quantitative methods based on modeling and simulation are here to help, and they can both help to understand the data, already generated data, and help to plan the clinical studies and preclinical experiments as well. So the quantitative methods in modeling in generic drug approvals are part of the concept which is called model-informed drug development. And in this case, it will be the combination of the model-informed drug development and something what is called model-integrated evidence. As you will notice, I am using some of the slides which were some of the material which was previously presented, because it's probably the best way to inform or to provide information about the topic which I want to present. So, what is this MIDD plus MIE, or model-informed drug development and model-integrated evidence in practice? In practice, it's an integration of existing knowledge with the use of mathematical models with verified predictive performance. And this predictive performance verification and quality assessment is probably one of the crucial elements, together with the proper information which describes the physicochemical characteristic of the drug and information about the formulation. It is utilized for intended modeling purpose. And this is probably extremely important. Only information that is a combination of science and knowledge and is sufficiently supported, validated, and verified can be classified as model-integrated evidence, with the endpoint, which is a regulatory decision-making. Having said that, the same approach can be used internally for decision-making. The same approach can be used to optimize the clinical trial. The same approach can be used to optimize and plan the preclinical research as well. So it's not only about the regulatory decision making. It's not necessarily the highest possible level. The same approach can be used earlier as well. Whenever we have new data coming from the experiments which we are running in the labs, coming from the clinical trials, coming from the knowledge which we got about the drug, modeling and simulation and in silico mathematical modeling and simulation can help to integrate this evidence. And just as an example, this model integrated evidence can be used to plan a pivotal study, but at the same time, it can reach another level and serve as a pivotal study itself. So combination of the preclinical data plus knowledge integrated or analyzed with use of the model can be used as a surrogate of the clinical trial. I'm mentioning model-informed drug discovery and development. And I'm using the term modeling and simulation, but what I specifically have in mind, at least for this presentation, are physiologically based pharmacokinetic models. It doesn't mean that that's the only tool which can be used. There are other modeling techniques which can be utilized for this need, just to mention population pharmacokinetics. But in general, PBPK models are perfectly suited to deal with this kind of studies. So the combination of the preclinical data, information about the formulation, information about the population variability are all covered by this approach. What is the best practice of the PBPK model development and verification? It's a process. So it's not like a one step. We have a model and we apply the model. It is something what takes time, and it is like a circle. So we are developing the model, we're assessing the quality of the model, then we're coming back to the starting point. We're assessing whether the model is good enough, what is the uncertainty of the model, what are the parameters which needs to be modified, or what is the data even measured, which is probably not optimal, or there are some problems connected with this single parameter. The model helps us to understand really the problem. But whenever we have a model which we are happy with, there is a next step and utilization of the model. In this case, it's virtual bioequivalence analysis, and then the issue is to analyze the data properly and to report, to document and report the outputs. As you probably perfectly know, PBPK is not a new concept. It's been defined 100 years ago. That's something what is repeated during each and every presentation, so I'm not going to go to that. However, this slide and this cartoon, which was prepared about 10 years ago by Dr. Ping Zhao, is ideal exemplification of what I have in mind regarding the combination of intrinsic and extrinsic factors influencing the behavior of the drug, and how they should be combined with the mathematical structure of the model. That's the one thing. And second thing is how to, in practice, apply the predict, learn, confirm approach. So what I would like to draw your attention to is something what is connected with the intrinsic variability. So disease, gender, age, genetics, organ dysfunction, all these parameters can influence the behavior of the drug. And at least for some of the drugs, given topically, these parameters can be of significant interest, because they can just, simply speaking, influence the pharmacokinetics to the level which needs to be considered. It would be probably unethical and definitely economically not the best idea to run all these clinical trials for all these populations, and properly verified PBPK model, which combines variability, is perfectly suited to serve as a surrogate for this kind of trial. PBPK models have been applied very widely for different types of drugs and for different types of problems, beginning from the product-specific guidance development, new drug, new ethical drug developments, drug-drug interaction assessments, but at the same time, they can be used as an alternative for demonstrating bioequivalence. And again, the character and structure of the model makes it perfectly suited to deal with this kind of the challenges. There's one element which I want to mention as well. It's not the main part of this webinar and this presentation, but PBPK models have been recently applied for toxin safety assessment as well. And again, this is something what can be covered as a important element of the drug development by this type of modeling. I'm focusing on the dermal drug, both topical and transdermal, but in general locally acting. And I will describe a model which we use for this purpose, but it doesn't mean that it's limited to the locally acting drugs which are applied topically only. The same approach with different model and different physiological data can be used for all other type of drugs, including nasal, gut or inhaled, or gut dosed or inhaled. It's all about development of the model and verification, proper verification of the model. Whenever we have a model in place like that, properly verified, properly developed and verified, they can be used for different type of drugs as well. So the model which I have in mind and the model which I would like to describe with a bit more details is a model which we developed internally at Certara, Simcyp, starting a couple of years ago. The model is called MPML, which stands for multi-phase, multi-layer MechDermA model. It is a mechanistic model which describes the permeation of the drug through the human skin. The model consists of multiple elements and allows precise assessment of the changing concentration as the drug permeates down from the surface of the skin to the blood, to the systemic circulation. So the first element is a drug formulation, and I'm not going to spend much time on that right now because I'll come back to that. But that element, or this element, from the virtual bioequivalence point of view, will be absolutely critical. The model currently covers all main types of formulations, beginning from relatively simple, like simulation, solution, through emulsions, suspension, and patches, and proper characterization of the formulation will be absolutely critical for the virtual bioequivalence analysis. The first element, which is physiological and describes the physiology of the skin, is obviously stratum corneum. For the stratum corneum, what do we need? We need to be able to track the concentration of the drug as it permeates down towards viable epidermis. There are two options how to deal with that. The first one is to build a spatiotemporal model and to account for the permeation not only in time but in space as well. But for that, we need partial differential equations, and that makes potentially the simulation pretty long and inefficient. The other idea is to divide the stratum corneum to sublayers, and with use of the ordinary differential equations, describe each and every layer. And probably in this approach, the biggest challenge is to describe how the drug permeates, how the drug partitions between different parts, and that's something what is important element of this model. As you know, stratum corneum has a specific structure. It's called brick and mortar type of structure. So one of the elements which we have to account for is tortuosity, because most of the drug will permeate through the lipidic mortar, and therefore the permeation won't be linear, and the tortuosity can be potentially important parameter. Then there are deeper parts of the skin, beginning from the viable epidermis, dermis, up to the subcutaneous tissues like subcutis or deeper tissues like joints or synovial fluid or muscle. So the model has been parameterized with use of the human data and contains information describing all of these structures. The default deep tissue structure or deep tissue is muscle, but it can be reparameterized to describe different tissues of interest, like synovial fluid, for example. I specifically left a left-hand side free to say a few words about the new model which we recently developed and introduced or implemented to Simcyp Simulator, which is called the RDM, or multi-compartmental dermis model. Why? Because at least for some of the drugs, permeation through the dermis, and more importantly, clearance triggered by the blood flow, or maybe for some of the drugs, lymph flow, can be of interest and can be a critical factor. To be able to account for that mechanistically, we developed a more complex model as compared to the original one, which was assumed to be well-stirred single compartmental dermis model. It allows us to track the concentration as the drug permeates through the dermis, and for the drugs which act locally in the dermis to assess precisely local tissue concentration. One of the most important elements of the model is variability, which is connected with the skin. And for that, we develop multiple populations which are parameterized with the data coming from the clinical trials or clinical observations. The default population is obviously a healthy population, healthy skin. But apart from that, there are some special populations, including disease populations. So it is pediatric, it's elderly skin or elderly populations with certain differences as compared against adults, healthy adults structure. But probably most importantly, we have a disease population as well. I'm just giving one example here, which is psoriasis. And the structure of the skin is modified to the level which can really influence the behavior of the drug in the skin. That's something what is part of the model. And apart from that, the model is connected with the whole Simcyp machinery, so we can choose either full or minimal PBPK model, and to assess not only the local tissue concentration, but the systemic concentration as well. I mentioned formulation, and I said that it is of critical importance, especially for the assessment of the virtual bioequivalence. As I said previously, all four main types of formulations are covered by the model. And for each and every of this type of formulation, the user is supposed to provide information which describe the formulation of interest. So beginning from the probably simplest one, which is solution, where the most important factor will be the diffusion and partition between the formulation and certain colonial lipids. The more complex formulation we have, the more information we have to provide. So for example, if it is suspension and we have a solid particles or solid phase as part of the formulation, we do have to provide information about that. So what is the number of the particles? What is the character of the particles? With that, what will be the dissolution of the drug from the particle? And all of that will be accounted for during the simulation. From our experience, for majority of drugs which are given topically, evaporation and with that precipitation as well will be of critical importance. So whenever the vehicle evaporates, it changes the formulation to the level which has its consequences further on, and changes the behavior of the drug, both at the skin surface and deeper in the skin. So we do have to account for that, and we do have to provide this kind of information to be able to parameterize the model. Apart from that, other important parameters include vehicle pH or formulation pH, because depending on that, the ionization at the surface of the skin will be different. I spent quite a lot of time on that by purpose, because I just want to clearly say that this critical quality attributes are really critical, and proper parameterization or providing proper information, correct information, which describe the formulation is of critical importance. And that's something that cannot be forgotten, especially for the virtual bioequivalence analysis. Apart from the model itself, apart from the variability, which comes from different populations, from different locations at the body, we cover 8 of them at the moment, as part of the model. Apart from the formulation-specific parameters, there are other elements which are important, and this step is really important step in model development and model building. This includes the area of application, thickness of the applied skin, number of individuals, and all that. there's one thing which I want to quickly discuss here. So I just want to kind of use this slide as an excuse to discuss one of the elements which is of critical importance, which is a structure of the clinical trial, or which clinical trial schema we follow. There are two ways how to do it. Obviously, the first one is to run parallel study, and the other one is crossover study. With the crossover study, the important parameter, or the critical parameter, is between occasion variability. And that's something what is pretty challenging to cover, actually. And to my knowledge, there is no well-known tool or approach to address this challenge at the moment. I'm not trying to say that it's not possible. It is, but it's pretty challenging. And actually running the simulation for a proper, correct simulation for the crossover study is extremely challenging, because we do have to account for this between occasion variability. The same individual on Monday and 1 week later, still Monday, but 1 week later, will be potentially different enough that we should account for that in our simulation. How to do that? It's not such an easy task. At the same time, parallel trials offer a solution for this problem because of the nature of this study. So if the population is large enough, we can reach a certain power of the study, and we can cover this between subject variability properly with this kind of clinical trial, or this structure of the clinical trial. I mentioned previously that virtual bioequivalence approach, for example, with use of the MPML MechDermA model, can be used. And I started with the information about the recent success. And this is exactly how it was done. So it was used as an alternative approach in combination with Q1, Q2, and Q3 information. And the Dermal PBPK model was used in lieu of an in vivo comparative clinical endpoint study. The model development included characterization of the API, API ADME properties, and, as I mentioned previously, information about the formulation attributes for the drug product, for test and reference drug product. This included a series of parameters, physical and physicochemical parameters, including viscosity, pH, and others, which were used to parameterize the model and to analyze the differences between the reference and test, and allowed later on to analyze the bioequivalence of this drug. Having said that, it's probably not, it is for sure not the only potential use of PBPK in general. It can be used earlier to inform candidate selection. It can help to translate from animal to human, and so far and so on. So for all of that, PBPK models are extremely useful. With that, I would like to give you one example where dermal model, in this case MPML MechDermA, can be used for innovative drugs. So it's a case study which was presented by colleagues from Galderma during the APS webinar earlier last year. And in this case, MechDermA model was used to support approval for active cream. And there were two types of questions asked during the drug development. The first one was the risk connected with the potential drug-drug interaction. And the other one was whether we were able to translate the result, the clinical results, or combination of preclinical and clinical results generated for adults to the pediatric population. And for that, the proper model or model was developed to describe triparatin ADME properties based on the in vitro metabolism data and with information which describe the nature of the formulation. And the model was used to run virtual drug-drug interaction study with inhibition of the main enzyme involved in the tripharotan metabolism. In this case, it was 2C9. So the results informed or gave the information without running the clinical trial that even if we have increase in the systemic exposure, in this case, an average of 50%, it is still within a safety margin. So in this case, the clinical trial was not necessary, and the model informed scientists, researchers, that we are still within the safety margin. Again, it included the whole model building procedure, model analysis, and quality verification step before it can be reported. But then whenever we have a model which meets all the criteria, the quality criteria, we can use such model to run this kind of a clinical trial as well, a virtual clinical trial. The other element, which is probably one of the largest benefits of the proper PBPK model, or properly developed PBPK model, is its ability to translate the results from one population to another. So what we have here is the information, or clinical information about the concentration, measured concentration, after multiple doses of the drug given topically for the 12 to 17 years old individuals. But at the same time, the clinical question is whether we can expect the same systemic exposure for younger individuals, in this case, 9 to 11 years old kids. So what was done, my colleague, Dr. Patel, developed in collaboration with colleagues from Galderma, developed a PBPK model, which was then verified with use of the clinical data for teenagers from 12 to 17 years old. And then the simulation was run for younger individuals to assess whether there will be clinically significant difference in the exposure for younger individuals. And there are two conclusions and two observations, actually, which can be made out of this graph and out of this study. So the first one is, which is probably clinically most important, there is no big difference and there is no significant differences for the exposure. So the safety is not jeopardized. So there is no problem from the safety perspective. But there is other conclusion as well. One of the limitation, or one of the challenge which we face when we run a clinical trial or any type of the research is something what is connected with the analytical method development, and how to deal with the LOQ and the measurements which are below level of quantification. PBPK model have no such problems, in fact, because what we do, we report as an output just concentration. There is no LOQ for PBPK model. So knowing LOQ, or by replacing LOQ by safety threshold or efficacy threshold or any other threshold which is known from previous clinical trials, we can use the model to compare the predicted concentration versus this threshold and to draw some conclusions out of it. And this is exactly the case in this case study as well. I quickly run through two case studies or two trials which we're involved in, but of course it doesn't cover everything of what we're involved. And Simcyp during the last 20 years has a significant impact on the novel and generic drug labeling. So we've got more than 200 label claims using PBPK in lieu of clinical trials, and some of the drugs are presented on the screen. What proves that with properly developed and verified PBPK model, we can tackle really different clinical questions, and really various problems can be tackled and potentially solved. With that, I would like to encourage you to contact us if there are any questions, if there's any idea of a collaboration. We'll be more than happy to be in touch and collaborate with you. So what are the main take-home messages? First of all, PBPK model, the dermal PBPK model, MPML MechDermA model is in place, and it can help sponsors to save time and money developing novel and generic dermal drugs. General observation, in silico model and simulation minimizes exposure of children and other sensitive populations to experimental drugs. And once again, I would like to mention this variability between subject variability and between population variability. This is of extreme importance, and I guess it will be more important when we go farther with the novel concepts. We don't want to unnecessarily expose this population to the drugs. With that, why not to use properly developed PBPK models? Virtual bioequivalence is here. It's not without challenges. One of the challenges which I mentioned is the study character, whether it is crossover or parallel. There are more challenges, more issues, but in general, the strategy is here, the framework is here, and the framework can really help to access to complex generic drugs, and it has regulatory backing at the moment. The last one will be, please stay healthy, and I'm 100% sure that we as a humanity will deal with this new virus. I'll be happy to take any question. Thank you very much.
Thanks so much, Bastek. As Dr. Pollock mentioned, he'd be happy to take your questions. Please enter them into the Q&A box, and we'll try to answer as many as we can in the time allotted. So it looks like we've got a couple questions from the audience already. First one is, someone says, stratum corneum may not be considered a significant rate-limiting factor in drug absorptions, but other skin layers may not have significant impact. For modeling purposes, do you need to consider skin components other than stratum corneum?
My answer is yes, because it's an interplay and combination of multiple factors. Depending on the formulation or depending on the character of the API, it can change. So, for example, if you have a drug which is metabolized inside the skin, even in the stratum corneum, because it can be some degree metabolically active, the parent can differ, can behave differently as compared against the metabolite. So, for example, for the parent, the stratum corneum will be a barrier, which is not necessarily the case for the metabolite. With that, I would say yes, we do have to account for all these elements. Having said that, PBPK model can help us to find these critical elements and this rate limiting factor. It's not necessarily stratum corneum. If it is not, PBPK model can help us to find this and maybe to develop a lumped model, simpler model, which will answer our question. But in general, at the beginning, I would probably prefer to have the whole machinery in place and to analyze the problem carefully from the beginning.
Got another question. It sounds like you may have answered this, but you may want to add a little bit more detail. Someone asks, does the MechDermA model take into account any metabolism within the skin layers?
Yes, it does, but not in a mechanistic way, at least at this stage. So that's something that we are working on, and that's one of the logical extension of the model. So far, yes, you can define the metabolism in the metabolically active, most metabolically active part of the skin, like via viable epidermis and dermis, by defining the clearance. But as I said, it's not fully mechanistic.
Someone wants to know whether they can enter in vitro skin model data directly into the MechDermA model to make meaningful decisions.
In vitro data, which come, for example, from the IVPT study or IVRT study, can be used to parameterize the model. You don't directly input results of this study, like, I don't know, flux, for example, but you can use this experiment and the data from this experiment to parameterize the model. So the answer is yes, you can, but not necessarily directly. Having said that, we find both of these studies, IVRT and IVPT, extremely important. And MechDermA model allows you not only to use this data, but also simulate the IVPT itself. With that, you can optimize the IVPT study before running it. So that's another use of MechDermA model.
Can the MechDermA model be used to predict drug disposition after subcutaneous or intramuscular injections?
MechDermA model itself, no. It's been designed to deal with the topical application of the drug. Having said that, we do have a separate model for subcutaneous injection, and intramuscular injection is on our radar as well.
As you know, the density of hair on the skin varies, so it's not very dense on the hand, but it's quite dense on your head, for most people at least. Can hair shafts act as a shortcut for drug absorption straight into the dermis?
The answer is yes. Thank you very much for this question. I should have mentioned that previously when I was describing the model itself. Yes, we do account for the skin appendages. In this case, hair shafts is like a shortcut for the drug, and allows the drug to be taken from the surface of the skin and dumped directly to the dermis. For at least some of the compounds and some of the formulations, it can be important. And we've got at least 2 clinical trials and 2 case studies for which we showed that the model can differentiate the situation with and without hair or hair shafts in the skin. One of them was nicotine. And in general, model can deal with that. And the other part of this question is the variability. That's a very good point, and we do account for that. For different locations, we've got different density and character of the shafts, hair shafts. So we do account for this between location variability as part of the model.
This question may need some clarification. So someone says, to show bioequivalence of completely different formulations, which IVIVC validated methods are required to use PBPK modeling besides the formulation's critical quality attributes?
Oh, well, yes, I agree. This question is like a beginning of a one-day-long discussion, so I won't be able to answer it quickly. Just to try to tackle most important elements of this question. First of all, IVIVC, in my opinion, is not absolutely necessary for virtual bioequivalence or for the bioequivalence in general. It is extremely useful, and it should be done, but it's not absolutely necessary. That's the one thing. The other thing is, and it's probably more important, whether we can use this approach to deal with the drugs which are not Q1, Q2, Q3, or we've got non-Q1, Q2, Q3 formulation or different formulation. In my opinion, yes, but it's only my opinion at the moment. It adds to the complexity, or it makes the problem even more challenging. But this is exactly what PBPK model is for. As long as we have a verified model, but non-identical formulations or even different formulations, PBPK model can describe which parameter influences the behavior of the drug and whether these differences between different formulations are significant enough to make this drug clinically non-bioequivalent. So that's a very quick answer, but as I said, that's something what we could probably discuss over hours. Very good question, by the way. Thank you.
Someone wants to know whether the MechDermA model is able to take ethnic differences into account.
Yes, that's a very good question. Thank you very much. Yes, it can. At the moment, we do have two healthy volunteers populations for two different ethnic groups. So we've got North European Caucasian population and we've got East Asian population. We're not able to differentiate between Chinese, Korean, Japanese, but in general, all the reports suggested that the skin is different in North European Caucasians and East Asian in the populations, but there's no significant difference as for the skin structure between these three different groups. So we developed one combined lumped population, which is called East Asian population, and structure or description of the skin is connected with all populations which we have at the moment in Simcyp, which is Chinese population and Japanese population. Yes, the machinery is there, and the data is there. More important.
Someone wants to know, when talking about deep tissue, do you have to choose between synovial fluid and the joint?
It depends on the question which is asked. Because at the moment, as I said, the model is parameterized by default for the muscle. So we do take responsibility for the muscle, and the muscle is the default tissue. However, as I said previously, it can be parameterized to describe other deep tissues. From our experience, we did it for the synovial fluid, and the prediction was really good, and it can be done. Having said that, I would say that if we want to have a mechanistic model in place, which describes the joint in general, it should be described with all the details, which builds the complexity of this tissue.
Okay, hi. Someone wants to know whether you can confirm any correlations between actual observed time concentration profiles versus those predicted by this PBPK model for historical topical transdermal products. If there's not a good correlation, what would be the main reason for that?
Oh, that's a very general question, and I'll probably revert the question, and I would say that PBPK model is perfectly suited to understand the problem or source of the problem. And yes, I agree. It's pretty common that we have, we build model relatively quickly just to see whether we are in a same ballpark range as compared against the clinical observation, and the prediction is, well, not perfect. So now we have a model which allows us to answer the question, what is the reason for that? And we do that in a very systematic way. We analyze parameter by parameter, checking what is the source of the problem. And surprisingly, I was surprised when I saw it. We've been recently working, one of my colleagues have been working on a compound, which is very well known. It seems that we've got a full set of data to describe this compound. But then we realized that the log P, probably the simplest parameter which you can think of, is of critical importance. And we believe that the measurement which was done not properly. And it was further confirmed because we had a chance to have a different log P data and a different source of the log P. And then the model was able to predict properly. And what I'm trying to say is use the model to understand the problem. That's exactly how PBPK model can be used. Sometimes it will be formulation. Sometimes it will be API even. I don't think it's that important as for the other parameters. Having said that, I hesitated because an hour ago we discussed a similar problem, and we had two different the same formulation applied for two different locations, like face and back, and the differences were really large. So sometimes the physiology can be a source of the problem or wrong prediction if you don't define properly the clinical trial settings. So actually, that's the hint, and that's what I would like to advise. Use the model by analyzing each and every parameter one by one to understand what is the source of the misprediction.
All right. Got a few more questions. Some people that you might want to model, for using MechDerma may have a skin disease that would cause their skin to be damaged and broken. Does MechDerma take skin disease into account?
Yes, it does. So one of the populations, one of the disease which we described is psoriasis, and this is exactly the case. There are cracks in the subconium, and subconium is modified to the level which modifies the absorption of the drug and permeation of the drug. So we do account for that. We do account for these cracks. We do account for the modification of the skin. Having said that, it will be the combination of different factors at the same time, because cracks create like a shafts, and the drug can reach deeper part of the skin quicker. But at the same time, the subconium is thicker. With that, it will be the interplay between these two different parameters. And depending on the formulation, and depending on the API, the exposure can be either higher or lower. So again, physiology will be one part, but it should be connected with other elements as well. Short answer is yes, we do account for that. And as an example, we have a psoriasis population.
Can you give some comments about the MechDerma pediatrics population?
Yes. So it's one of the population, and it is of extreme importance for us. We do account for the ontology, so age-dependent modification of the structure of the skin. So as the kid grows, the structure of the skin changes. So the thickness will be different. The pH will change, as long as other parameters. And running the simulation for different age groups, you can observe how each and every parameter and its modification in time influences the behavior of the drug. So yes, it is part of the system right now, and it's been verified with multiple drugs.
How does the MechDerma model deal with high drug variability? In this situation, the FDA recommends using a crossover replicate trial design, but this design is very challenging for bioequivalence, to demonstrate bioequivalence. So they were hoping that you could comment on this.
Yeah, that's a very good question, and it's very challenging. To be perfectly honest with you, I've never dealt with a problem of highly variable drug given topically. I'm sure there are some, but I never had a chance to work with them. Running crossover clinical trial or crossover virtual clinical trial is possible. The biggest challenge, as I mentioned previously, is a proper parameterization of the between occasion variability. There is no clear idea how to do that. There is no clear paradigm or method how to do that, because there are multiple parameters or multiple elements which influence this between occasion variability. It is theoretically doable, yet very challenging. If we want to have it done properly, obviously, not running the same population and calling it crossover. It's not crossover.
Does the MechDerma PBPK skin model consider the impact of external heat or water on drug absorption?
It can be done indirectly, so it's not part of the model itself. We do not have a mechanistic description, mechanistic mathematical description of the heat and how the heat influences the behavior of the drug, but it can be done indirectly. So if you have this kind of data measured in vitro, this is something that can be applied to the model. As for the hydration, yes, hydration is one of the physiological parameters which we use to describe the skin or subcutaneous, to be more precise. We have depth differences. So for different depth of the subcutaneous, the hydration will be different, and we can account for modification of these parameters. For example, psoriasis patients have modified or different water content as compared against the healthy skin. Again, this is something what we have to account for, and we do.
Looks like we have one final question from our audience. Someone says that they want to know about the model for synovial fluid, and can this model predict either plasma concentration or local drug concentration in the synovial fluid? And then the second part is, could you define the relationship between plasma concentration and local efficacy of the drug injected into the joint?
Okay, so there are at least three questions in one question. I'll try to quickly address all of them. Yes, the model can be used to predict local tissue concentration, in this case, synovial fluid, although you do have to provide the physiological parameters which describes the synovial cavity, volume, structure, density, viscosity of the synovial fluid, and all these kind of things. The drug is not directly injected to the synovial fluid. It's applied topically, then permeates through the skin and appears in the synovial fluid. And the interplay between the synovial fluid concentration and plasma concentration is an effect of the interplay of all those physiological and drug-related parameters. So I can't give you a direct answer for that it will be of a factor of 3, 5, or 15, because it will depend on multiple factors, really. But in general, yes, it is doable, and it can be done, even currently with the current structure of the model.
Looks like we've got one final question. Someone wants to know whether the model can take into account different vehicles or delivery systems used to carry the drug, or whether it only looks at the pure API.
Oh no, it's not pure API. If that was the that was the impression, it's no. Yes, definitely, and it's part of the characterization of the formulation. So yes, to give you a short answer, definitely the model can deal with very different vehicles or mixtures of vehicles, because it's very rarely a single excipient, and this will be characterized at the level of the solubility of the API in the vehicle or vehicles, molecular volume, viscosity, all these parameters which needs to be provided to parameterize the model.
Great. Thank you so much, Bastek, and thank you so much to our audience for all those great questions.
Once again, thank you very much. It was a great pleasure. Thank you.
Before concluding the webinar, we have a few short announcements. You can register for all webinars in the series by visiting certara.com/webinars. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.